Biochemistry Part 2. Organic compounds are compounds that contain carbon. There are 4 classes of organic compounds: carbohydrates, lipids, proteins, and nucleic acids. Carbohydrates consist of only 3 elements: carbon, hydrogen, and oxygen. Carbohydrates supply quick energy. Examples are rice, pasta, bread, and cookies. There are 3 classes of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Monosaccharides have a chemical formula of C6H12O6. Examples are glucose, galactose, and fructose, which are all isomers of each other. Isomers are compounds with the same molecular formula but with different structures. Therefore, they have different physical and chemical properties. Disaccharides have the chemical formula C12H22O11. They consist of 2 monosaccharides joined together by a process known as dehydration synthesis. So realize, since monosaccharides is C6H12O6 and disaccharides are C12H22O11, it's 2 times a monosaccharide minus a H2O. Examples of disaccharides are maltose, lactose, and sucrose. Hydrolysis is the opposite of dehydration synthesis. It is the breakdown of a compound with the addition of water. It is what occurs during digestion and is the reverse of dehydration synthesis. So in dehydration synthesis, the byproduct is water. But in hydrolysis, they use water to break down the disaccharide. Polysaccharides are polymers of carbohydrates. They form as many monosaccharides are joined together by dehydration synthesis. Examples are cellulose, starch, chitin, and glycogen. Cellulose makes up plant cell walls. Starch is the way plants store carbohydrates. Chitin makes up the exoskeleton in arthropods and cell walls in mushrooms. And glycogen is known as animal starch, which is stored in the liver and skeletal muscle. Cellulose and starch is found in plants, and chitin and glycogen are found in animals. Lipids are a diverse class of organic compounds that include fats, oils, and waxes. Structurally, most lipids consist of 1 glycerol and 3 fatty acids. Glycerol is an alcohol whose structure consists of CH and O's. A fatty acid is a hydrocarbon chain with a carboxyl group at one end. Fatty acids exist in 2 varieties: saturated and unsaturated. Saturated fats, with a few exceptions, comes from animals. They are solid at room temperature and when ingested in large quantities are linked to heart disease. An example of a saturated fat is butter. Saturated fatty acids contain only single bonds between carbon atoms. Unsaturated fats are extracted from plants, are liquid at room temperature, and are considered the good dietary fats. Unsaturated fatty acids have at least 1 double bond between carbon atoms in the hydrocarbon chain. Thus, they have fewer hydrogen atoms. So a good way to remember this is saturated means it's filled with something and it's filled with as many hydrogens as possible. But in unsaturated fats, Since there are double bonds, there are fewer hydrogen atoms. Lipid functions include storing energy, structure, like phospholipids are a major component of the cell membrane, and endocrine, which are some lipids are hormones. Proteins. Proteins are polymers or polypeptides consisting of repeating units called amino acids, joined by peptide bonds. Amino acids consist of a carboxyl group, an amino group, and a variable R, all attached to a central carbon atom. The R group or variable differs with each amino acid. Proteins are responsible for growth and repair. They include fish, meat, and beans. Proteins consist of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. With only 20 different amino acids, cells can build thousands of different proteins. Enzymes are examples of proteins, and two amino acids combine to form a dipeptide. A dipeptide is a molecule consisting of two amino acids connected by one peptide bond. Protein structure. Proteins have many functions in living things. They act as enzymes, membrane channels, and hormones. The function of the protein depends on its shape. The shape of a protein is the result of 4 levels of structure: primary, secondary, tertiary, and quaternary. Primary structure results from the sequence of amino acids that make up the protein chain. Secondary structure results from the hydrogen bonding within the molecule. The helical nature of many proteins is a result of hydrogen bonding. Tertiary structure is the intricate three-dimensional shape or conformation of a protein and most directly determines the way it functions and its specificity. Enzymes denature, which means they lose their natural shape in high temperatures or adverse pH. When a protein or enzyme denatures, it cannot function because its tertiary structure has been altered beyond repair. Quaternary structure refers to proteins that consist of more than one polypeptide chain. So, remember, tertiary structure is directly responsible for the shape of a protein and how it functions. And primary structure is a sequence of amino acids and is a chain. Secondary structure is hydrogen bonding and can be helical. And tertiary is three-dimensional shape. And quaternary is when there are more than one polypeptide chain. Enzymes. Enzymes are large proteins. Enzymes serve to speed up reactions by lowering the energy of activation, the amount of energy needed to begin a reaction. The chemical that an enzyme works on is called a substrate. Enzymes are specific. For example, only substrate A will bind to the enzyme. The induced fit model describes how enzymes work. As the substrate enters the active site, it induces the enzyme to alter its shape slightly so the substrate fits better. You can kind of think of it as a lock and key model, but the substrate would change. Enzymes are not degraded during a reaction, but are reused. Enzymes are named after their substrate with the ase suffix at the end. For example, lactase, sucrase. Enzymes function with the assistance from cofactors, which are minerals, or coenzymes, which are vitamins. The efficiency of the enzyme is affected by temperature and pH. As enzymes denature, they lose their unique shape and their ability to function. Prions. Prions are infectious proteins that cause several brain diseases, including mad cow disease. A prion is a misfolded version of a protein normally found in the brains of mammals. If a prion gets into a normal brain, it causes all the normal proteins to misfold in the same way. Nucleic acids. The nucleic acids are deoxyribonucleic acid and ribonucleic acid RNA. They carry hereditary information. They are polymers, chains of repeating units of nucleotides. A single nucleotide consists of a phosphate, a 5-carbon sugar, either deoxyribose or ribose, and a nitrogenous base. Nitrogenous bases are adenine, cytosine, guanine, and thymine in DNA, and in RNA, instead of thymine, it's uracil. Adenine and guanine are purines. A way to remember that is angels have pure hearts, and the hearts refer to 2. And the cytosine and thymine and uracil are pyrimidines.